Just keep in mind as I'm speaking that Yanni, who's fortunately speaking right after me, will be presenting some really neat tools that we discussed yesterday. So when you're watching what I'm talking about, and when you're watching what he's talking about, just think, I'm going to go ask Yanni to help me make these tools even more exciting. OK, so I am kind of a half biologist, half computer scientist. And I'm very interested in applications in biology. And I'm interested in these tools partly because they're cool, but partly because I'm interested in having impact in biology. So that's just the context for my talk. And I'm going to talk about biomolecular networks that come from single-cell data. These are 3 cells that are undergoing apoptosis. And there you can see them in 3 different stages. Apoptosis is cell death, when the cell chooses to destroy itself in order, for example, to protect us from cancer. So a little bit of biology for people who hasn't taken biology? A bunch of people, right? OK, so just this is biology in one slide. All you need to know about biology in one slide. So this is a B lymphocyte. It's a part of your immune system. Without it, you will die. On its surface are proteins called receptors. And all cells in your body have receptors. And they are to allow them to respond to their environment. So something will come from the blood, let's say, and will bind to this receptor. And the B cell will respond in such a way that it will save you from the virus or bacteria that's attacking you and save your life. All your cells are constantly responding to their environment every second of every day. And that's what's keeping you alive in I don't know, billions of different ways. So inside the cell, how does the cell respond to these things binding to its receptors? Actually, the receptor is this protein, and when the thing binds, it makes the protein shift, and that gives it different kinds of functionalities. And the way that's carried out, and when the cell carries out its functionalities, it might decide to die, it might decide to divide, it might decide to secrete something to talk to the other cells. How does it do that? Inside the cell, there's a network of proteins that carry out these different functionalities. And what I'm very interested in, is this network of proteins. And the reason I'm interested is that when this network of proteins misfires, is poorly regulated, then we can get diseases such as cancer. The cell decides to divide and divide and divide, or the cell decides to go somewhere, and that can lead to metastasis and so on. So we're very interested in these networks because they regulate what the cell can do, and they can lead to diseases. In biology, in molecular biology, still today, lysate-based data is the norm. What do I mean by lysate-based data? We get a test tube of cells, let's say from a cancer patient. We put it in a blender, essentially a lab blender, and we make a lysate of cells. And then, using different techniques, I'm showing a classic one called a Western blot here. Using different techniques, we fraction out the proteins and quantify them. What does that give me? So I started with, I don't know, 1 billion cells in this test tube. What do I get after I do this lysate-based method? I get a vector, 1 measurement for every variable of interest, 1 sad little measurement. So all I get is a vector. I can't ask questions on the multivariate scale, right, unless I get lots of patients. But for that 1 patient, I can't ask any questions even about simple correlations, because I only have 1 measurement. And in biology, generally, we'll have 2 kinds of conditions that we care about. We'll profile them using these lysate-based methods. And whatever it is that we care about, we'll say, aha, some protein went up and some protein went down. In the cancer patients that didn't respond, this protein was higher. In the cancer patients that did respond, this protein was lower, and so on. In contrast, flow cytometry data is single-cell analysis. So here's a crash course in flow cytometry. We have this multi-well format plate. Imagine there's multiple test tubes. It's just more convenient in a plate. In each one, we might have, for example, a different patient. And now I'm zooming in on 4 different cells. And 1 protein of interest I've labeled in blue, and 1 protein of interest I've labeled in green. And you can see that this cell has a lot of the blue and not much of the green. And this cell has much of whatever I'm tagging in green, and not much of whatever I'm tagging in blue, and so on. And something called flow cytometry is a technology that allows you to flow the cells through and quantify for every single cell how much it had of the different proteins. That's single-cell analysis.
Cell of the body or cell of
the cell that came from the test tube. Whatever I put into the machine, which in our case is usually blood cells or bone marrow cells. But whatever it is that you want, as long as it's in liquid form, which is sometimes a challenge. OK, so very recently, in our lab, we helped perfect a technology called mass cytometry. The problem with classic flow cytometry is that you could do, on a good day, you could do 10 or 12 variables. Most labs can do 4. So that's kind of, when you want to do multivariate analysis, 4 is very restricting. the signaling pathways might involve thousands of molecules. So mass cytometry, for reasons that Yanni will go into further, he said he would explain it, so I said, oh good, I have a pass. Mass cytometry, instead of using a fluorescent dye that you tag your proteins with, you tag them with metals, and then you use mass spec to identify the metal-tagged antibodies. Why do we care about that? Because we could do, out of the box like this, we could do 30. Today we can do a little bit more than 40. So that's amazing. For us, that was a huge leap in our ability. This is what the cytometer looks like. The newer one is not quite this as ugly a color. It's a nicer shade. For me, this was sort of the thing I did as a PhD student. The only important moment of my PhD was when I suddenly realized that single cell data, really if you treat each cell as an observation of the system, then wow, single cell data gives you high throughput, thousands of data points, just like that. You can run thousands of cells through the machine at each moment, yeah?
One basic question, isn't there always the issue with the peptides and you need to figure out how they map to the...
You mean the antibodies?
No, isn't it hard to measure proteins?
Oh, you are thinking of mass spec and proteomics. We've gotten around this problem. The way we get around this problem is we take antibodies and we attach them to metal tags.
Okay.
For the mass spec, it is trivial to quantify metal tags. Okay, so you're right. Usually mass spec and proteomics is very hard. For us, it's very easy. Okay? Good question. So the gist of this is that if you treat each cell as a single point, you have thousands of data points. Wow, that's amazing. That opens all kinds of doors for multivariate analysis. And this was really, I could have skipped the other years. That was the one interesting moment. OK, and the thing that you can do with this is you can treat the single cell data as high throughput data. Here are my single cells. I have some kind of flow measurement. For every cell, I quantify the relative abundance of different proteins of interest. And now I can say, OK, if I use some kind of PGM approach, can I find that underlying network? For every network in a molecular biology textbook, thousands of grad student hours went into figuring out this connectivity. Thousands. Can we do it in an hour on a cluster or in a few minutes on a laptop? So when you're talking about the spectrum of modeling tools that are available to us, this is sort of the clustering and so on you use when you really don't know anything about the domain. We'd love to make these dynamical systems models, but then you really need detailed information about the domain. So we're here in the PGM space, and we're hoping to be able to drag this over to the dynamical systems models once we've characterized the system very well. I won't talk about that, but that's one area of interest. Who here cannot explain to me what a Bayesian network is? Raise your hand if you don't know what a Bayesian network is.
You're too embarrassed by now.
No, but seriously, anyone? It's fine. I'll explain them in 2 slides. Anyone?
Okay.
Okay. Okay. So a Bayesian network. It's really a very quick explanation. A Bayesian network is a graph that looks like this, and each node here is representing, in my case, a protein, and an edge here represents a statistical dependence. So here I say that B is the child of A, and that means that B statistically depends upon A, and I'm hoping that maps to a causal relationship. I often cannot guarantee that, but I'm hoping. And what you can't see when you look at the Bayesian network is that underlying every edge is a quantitative description of how B depends upon A. Okay? So in this case, if A is on, B is in state 0 with this probability, in state 1 with this probability, and in state 2 with this probability. So it looks like A is an activator of B. That's all a Bayesian network is. Statistical dependencies and a quantitative description of how one depends upon the other.
Just to be absolutely clear, A is at the level of...
It's the relative abundance of the activated form of the protein.
Thank you.
Okay? So it's really, we're really getting into what we're interested in looking at.
Abundance relative to...
Because we cannot say how many protein molecules are there. Okay?
It's the probability that it'll be on.
No, it's we're quantifying how much we have of what we know to be the activated form.
With the Bayesian networks, we get probabilities, not numbers. So.
But the node is representing the relative abundance. Oh, how did I discretize? First of all, this is just a schematic to explain what the conditional probability distribution is. But yeah, we often do discretize. And how we discretize is another topic.
What is B probability over? Where's the probability of B over?
Probability that B is equal to, I've discretized.
Okay.
So this is just as cartoon,
it's
but.
not really frequent in the cell, and B is one is fairly
that the activated form is very low in the cell.
Got it.
Or it's off, or there's none. And here the activated form is in an intermediate state, and here there's a lot of the activated form. OK, so what does that mean? If this is single cell data, every dot here, sorry, they're not showing up, but every dot here is a cell. I have phospho A, I have phospho B. And you can clearly see that there's a strong statistical dependence here that will map to this edge in the Bayesian network. Importantly, if I look at the two proteins and I see no statistical dependence, don't forget that I've measured a whole bunch of proteins in every single cell. So I can also say, well, let's take into consideration protein E. And now I'm only going to color in red the cells that have high E. And suddenly a relationship emerges. OK? The point of this slide is to say Bayesian networks are not constrained to only pairwise relationships. All right, so we can find more complex, higher dimensional relationships. The other thing that's important is that Bayesian networks find what are called d-separators, which remove edges. Why do I care about that? Because I argue that the hairball approach to proteomic analysis is not that interesting. So to say, oh, everything here interacts with everything else, which is what everybody seems to say, is trivial and not interesting. I already know as a biologist that everything interacts with everything else. So I'd much rather be able to remove edges when I can. And the Bayesian networks are sophisticated enough to tell you that even though this guy is correlated with this guy, that even though everything is correlated with everything, this edge is unnecessary. And the way it does that is by saying, if I want to know the level of this, low, medium, high, let's say I've discretized, and I already know, will this guy help me predict it? Yes, it will, because it's correlated. But once I know this level, will I get any more information from this one? If the answer is no, no additional edge is put in there. OK, so that was a crash course on Bayesian networks. And the technical details. I'm sorry?
I thought it was due to Laplace. That's how
It's an inside joke.
I missed it, but.
Only Bayesians understand.
Markov too, right?
So, sorry, I have a ton of material, so I'm gonna rush through. So the technical details is that you can score a given graph. You can look at a given graph and say, how good is this graph structure given this data? You can score it if you have a nice form of the conditional probability distribution. And now we search for the high scoring model. Crash course in Bayes nets completed. Anyone have a question? OK, great. So this is what I did in my PhD. I took a data set from Gary Nolan's lab, which is where I am right now. They had done different perturbations of normal human blood. And they did 11-color flow cytometry, which was very impressive at the time. And they quantified these signaling molecules.
What are perturbations?
Oh, a perturbation is where you can set a variable to be on or off or some level.
Okay.
So you constrain the system.
Intervention, as we've been talking about.
Yes. So you have a data set of different conditions. These were primary human T cells, which is a type of immune cell. There were 11 variables in all, 9 conditions with 6 specific interventions, which of course we took advantage of. And there were 5,000 some odd data points, which was astonishing. Since no one had used single cell data in this way before, everybody was like, oh my God, how did you get so much data? So this is how we got so much data. And when we did that, we got an astonishingly accurate, I was shocked actually, an astonishingly accurate picture of T cell signaling. Again, thousands of grad student hours went into making this network. We made it, by running an algorithm.
How do you know exactly? Because if I pull your paper, the gold standard was based on literature search.
Oh, the gold standard is based on extremely well-documented networks. There are many connections in biology that you're not really sure about. Most of these are extremely well-documented. They're the bread and butter of biology.
Because there are some other ages in this pathway that I also know, but we're not.
True. So you can find an edge almost anywhere in biology, but most of these are extremely well known. So in biology, when you're asking about edges, something was done in the pancreas of a mouse, and something was done in the liver of a giraffe. I don't know what. I'm exaggerating, but it's very heterogeneous. But most of these are extremely well documented. They're the ones that you learn about in the college course. They're the ones in the textbooks.
So normatively, the word biology graduate student would go in and measure something about each cell and try to determine something about the structure of this network over many years?
Yeah, so there are many very difficult, cumbersome techniques in genetics and molecular biology that allow you to actually figure out these networks in a very sort of time-consuming way.
Right. So it's the same thing in certain brain circuits as well. So you know these circuits really well. That's what you're
Yeah. So I cannot guarantee you, first of all, surely we missed a lot of edges. And when we did works on cycles, we found a bunch of edges that aren't in here and that are probably true. And in biology, you never really know a gold standard. And yet, find me one biologist who will challenge, that RAF goes to mech. Never. Perhaps you would know the gold standard under a certain condition at a certain time point and so on. And yeah, you could do that in the particular.
Conditions.
Yeah. Okay. Let me, because this is not the most important part of the talk, let me say, as far as we could tell, it was astonishingly accurate. We didn't go through and check every single edge, okay? But many of these edges are extremely canonical, extremely well agreed upon.
Just for clarification, so when you talk about perturbation, can we think of that as an intervention in the causal DAG with that it's edge breaking?
Absolutely. Absolutely. The thing to remember, though, is that if I perturb MEC, I'm perturbing the activity of MEC, not the abundance of MEC. And I'm measuring the abundance of MEC. So that can be important.
Yeah, so the important difference, right, is that normally we cut edges going into a node, but you sometimes cut edges going out of it.
Yeah. So because the nature of these interventions, there are other interventions in molecular biology, but these are the easy ones. You just throw them in the test tube and you're done. And that's what they do. They block the activity of MEC, whereas the other ones are much, much harder to use.
And there's generally no feedback?
There is tons of feedback. I'll get to that. Yeah, but so yeah, we didn't find the feedback and we didn't find a lot of things. I mean, this is the dotted lines are missed edges and so on. I'm not saying it's perfect. I'm saying it's much, much, much more accurate than anything I expected to see, okay? And there was one that we didn't expect, that we weren't sure about, and we validated it using something called siRNA, which is a way of perturbing. And so we said, oh my God, this is great. We figured out how to find signaling pathways, except of course it wasn't that simple. So you might say, wait a minute. And this is what I said when I came to my postdoc. I was like, oh, wait a minute. It's not nearly as simple as I was hoping it would be. So first of all, what about the CPD? So the CPD, the conditional probability distribution, is the distribution that you use to quantitatively describe the connections in the graph, right? So what happens if you use a different CPD? Well, the answer is that nothing good. Things tend to be quite unstable. And I'll show you a slide on that in a second. Here's another issue. What about cycles? So we had a question about feedback. Absolutely. Biological systems are rife with feedback. So again, when I say astonishingly accurate, I just mean that it's much better than what I expected. I kind of expected things to be tossed on the page. And yet, can we actually expect these networks to reflect reality? No. We expect them to be very effective hypothesis generation tools. And when you interpret the model, you have to keep that in mind. You have to keep these problems in mind. Absolutely, things might be CPD dependent. Absolutely, the cycles are missed, and that can confuse things.
What do you mean by CPD dependent? I'm not sure.
So I'm going to use a linear Gaussian, and you're going to use a multinomial with discretization level 2.
With parameterization, right? So
we get different answers. I'm going to use a multinomial with 3 discretization states. You are too, but we discretize differently. Okay. Is that stable? I don't know. Sometimes yes, sometimes no. Okay?
What do you use to identify cycles?
Yeah, so this is an algorithm that I'll touch on very briefly, or you can look up the paper. But what about hidden variables? Hidden variables are sort of the bane of my existence. I was talking to Yanni about tools that sound really exciting to help alleviate some of these problems. He'll talk about them in a minute. What I've used so far is data integration. I have an example that I'm sure I won't get to. But you can, when you see an edge between A and D, it might be that A affects D, but there might be things in the middle. Or it might even be that there's a hidden variable that's affecting them both. And there's a lot of details in which one it might be. But I'm not going to get into that right now. Yanni will get into some of it. But I can look in the literature and say, has anything been reported for A and D? And like I was saying before, the literature is you know, the liver of the giraffe and the kidney or the brain cell of the mouse and everything is pooled together. So it's a mess. It's incredibly, incredibly noisy. But can it help me give some kind of evidence, some kind of support to what might be happening between A and D? Okay? So that's the idea there of the data integration. And finally, about dynamics, I'll touch on that as well. So here's the issue number 1 out of 4. What about, are things CPD dependent? This is just a quick slide to say, yes, they are. And here we've used a bunch of different CPDs. And here are different edges. And you can see that some of them are incredibly robust. And some of them are incredibly not robust. That's all I'm going to say about that. I just wanted to raise that issue. So if you want to be very thorough, you go through a bunch of CPDs. And that's what I typically do. But it's definitely a problem. OK, modeling cycles with Bayes nets. Anybody know how much time I have?
I do. You have about 15 minutes.
OK. All right, so modeling cycles with Bayes nets. This is published, so I'm just going to go through it very quickly. So we have the problem that there are cycles all over the place in biology. So if we want to accurately model pathways with cycles, what can we do? I'm just going to give you a schematic of what we do. So here is a molecule called PKC, and these are two histograms. This is a histogram where I stimulate the cells. I induce them to respond in some way. And this is a histogram where I do the same thing, and then I add a PKC inhibitor. Now, I'll remind you that a PKC inhibitor is something that blocks the activity of PKC. I'm not affecting the abundance of PKC, but I'm measuring the abundance of PKC. Why is this histogram shifted? Why should it be shifted? I haven't affected its abundance. The inhibitor is blocking its activity. And so I'm going to make the statement that if PKC affects PKC, I'm going to argue that there's a cycle from PKC back to PKC. Okay? And now I'm going to say, okay, well, what if I break that cycle open? Can I learn the cycle? Can I learn the network then? And so the outline of the algorithm is that here's, for example, a well-documented cycle in biology. What I'm going to do to try to learn it is I'm going to detect the cycles, like I showed in the previous slide. Now I'm going to break the cycles open with an inhibitor that blocks activity. So here, I'm going to break MEK here. And now I'm going to use the Bayesian network to learn the structure, which is now not cyclic. And then I can close the cycles. I won't get into how we close it, but you can talk to me afterwards if you're interested. It's basically with a conditional probability test. So here's a MEK inhibitor. It blocks MEK activity. Now the graph that we learn is going to look like this. And then we just need to close the cycle. So that's the sketch of the algorithm. And we went back to the T-cell data, and we said, could we find the cycles? And we could find the cycles that are kind of documented in the literature.
Can you go back one slide? How is it that when you inhibit MEK, you don't break the RAF to MEK edge?
Because I'm affecting MEK activity. I'm not affecting MEK abundance. So RAF is still phosphorylating MEK.
I see.
MEK abundance is still going up. So we went back to the T-cell data, and we said, oh, look, cool, we can find the networks now. I mean, we can find the cycles now. And we made some synthetic data. But as was pointed out, we never really know ground truth. And then we went and we made some synthetic data, and our results were perfect. And we said, well, yeah, I mean, we simulated everything very perfectly. Of course, the results are going to be perfect, right? So we're like, you know. So can we do better than that? And so we went to some friends of ours at Merrimack Pharmaceuticals. Merrimack Pharmaceuticals makes these ODE models, these dynamical systems models. And they make them, and they validate them, and they validate them. Because they literally make their drugs by using the predictions. And if you know something about the drug pipeline, you know that it's absurdly expensive. And so they have drugs in clinical trials based on these dynamical systems models. So these are as validated a model as you're going to find. And also they're friends of ours. So we could call them at midnight and say, help me, I don't understand something about the model. So here's the model. We perturb at these points. And you can see that there's a few cycles in there. And so we made synthetic data from this model. And we made as much as we wanted. We introduced a little bit of noise, but not too much. We had inhibitors. If you've ever worked in biology, you know that the inhibitors are a mess. And they say they're going to inhibit here, but they actually inhibit there. And they also inhibit 5 other places. And your life is hell when you're trying to model these. But when you're dealing with synthetic data, everything is perfect. Lovely. So then we did regular Bayesian learning, and we got this. All the dotted edges are wrong. And we used our cycle approach, and we said, it looks so much better. But then we scratched our head and we said, well, that's nice that it looks better. But why isn't it perfect? Why wouldn't it be perfect? I mean, what could be confounding our results? So my results are always confounded all over the place. Usually it's because there's cycles. Well, we took care of those. Or it's because there are hidden variables. Like I said, the bane of my existence. But there are no hidden variables. In fact, I called Jonathan and I said, are you sure there's no hidden variables? So what's going on? Was there insufficient data? No. We could use as much data as we wanted. And when we sampled down, we sampled up, we get the same amount, same answer. Were our inhibitors non-specific? No. The inhibitors are perfect. You could manipulate a synthetic system perfectly. Were the CPDs bad? I don't think so, because we tried a bunch of them and it was perfectly robust. This is very well-behaved, unimodal data. What's going on? We really banged our head against the wall. We couldn't understand why our results wouldn't be perfect. And what we came to understand is that the dynamics of the system, that's what we hypothesized, I should say, is the dynamics of the system are confounding the underlying structure. So the system is sort of in action. And you can argue that this should be steady state, but there's no steady state here. I mean, I would argue that there's no steady state here. You trigger the pathway to respond, it responds. There's just a degenerate steady state where nothing is happening anymore, and that's not very interesting. You can't learn anything because there are no dependencies there, right? So while something interesting is happening, the dynamics are in play. Oh, that was bad. Okay. And we have to appreciate that these are static models of dynamic systems. And in fact, when we simulated the differential equation model, what we did is say, OK, we're going to make thousands of initial conditions, different protein concentrations. And we think that mimics the biology as well as we can. And that's how and then we let it run for a while, right? So you're capturing these cells in sort of different states of their progression through this signaling pathway. And you have thousands of these cells, so it mimics the reality quite well. But the dynamics are happening, right? So this data is giving us a snapshot of the system behavior. Now, we looked in the literature, and the confounding effects of the dynamics had actually been previously described, not for static models, but in the context of dynamic models, in DBNs, dynamic Bayes nets, and CTBNs. They had already been described. And the word that they used was entanglement. So we kept the same word to make things clear. And I published just a tiny little snippet of this very recently. I didn't want to publish the whole story because I'm still trying to do the biological sort of demonstration. OK, so here are two examples of entanglement just to make it more clear in your mind. Let's say A is randomly set to 0 or 1 at time t. B is whatever A was a second ago. C is an or an and function on A of t and A of t minus 1. The real Bayes net should look like this. A affects B and A affects C. That's clear. But in reality, what's going to happen when I capture the data, the snapshot at time point t, what I'm going to see is this, right? B is capturing A's history better than A itself. In essence, A's history is a hidden variable. And B is representing that hidden variable better than A is. And therefore, you could very well get this structure. So that might not sound very biological. Here's a biological example. Let's say A affects B and C. And A takes a little bit of time to affect B and C. And let's say A is quite quick in changing. So it gets dephosphorylated, inactivated quite quickly. And let's say B has some intermediate rate of getting dephosphorylated. And C is quite slow. What's going to happen is that B is going to represent the activation level of A second ago better than A itself. And therefore, what we could very easily get is this kind of structure, which of course has a non-causal edge and is also missing a causal edge. So it's very non-optimal. OK, so here I have a bunch of sort of theoretical stuff. I'm just going to read off the titles, which translate the math into English, because I don't have a lot of time. So entanglement says that conditioning xi on xj of big T does not render it independent of xj of t minus little t. OK? So we're not independent of our history. And what we're going to do is formulate an algorithm that uses perturbations in a different way. Of course, we know in PGMs, especially in causal ones, that perturbations are everything that we need. They're very, very, very helpful in establishing causality. Here, we're going to use them in a very distinct sort of way. And I'll go through it in a second. So perturbations fix the value of xi to some value. They fix it in time because they perturb it.
Do they hold it there, or do they just put it there and let it go?
They hold it there because you leave the perturbation in the media. If you take the perturbation out, it will go again. And that can sometimes be important. And so I'm going to argue that perturbations will not introduce new instances of entanglement. And I'm just going to kind of speed through this. Any entanglement that was there after a perturbation was there before the perturbation. And where I'm going with this is that I'm going to use the perturbations to help me reduce entanglement. And we made a name for this. We're going to wrestle out the entanglement, haha. Reduced entanglement structure learning. So we're arguing that the perturbed variable is constant over time, and therefore it cannot cause entanglement. And so we're going to take advantage of the perturbations to uncover the correct causal edges and eliminate the non-causal edges. And just a very, very sort of high-level sketch of the algorithm as we're going to use it is that we're going to take a very perturbed condition. Let's say 4 variables are fixed at one time. And we're going to learn the causal structure. And now it's going to be incomplete because we have 4 inhibitors there. So there's a bunch of stuff missing. That's OK. We're going to argue that what we find, what we learn, we're going to trust more because that's less entangled. And now we're going to do structure learning. And we're going to enforce as a constraint what we learned under the high perturbed, less entangled condition. And when we did that, we got much less confounded results. I'm not going to go into the details unless you guys are interested. This turns out to be a causal edge, even though there's no equation. Jonathan, are you sure there's no equation? There's no equation, but it turns out to be actually a causal edge. And it's interesting, actually, but I'll skip it unless we have time at the end. So we ended up with one non-causal edge. This should really come out of here, the doubly phosphorylated form of ERK. And I don't know why. Maybe we didn't get rid of all the entanglement. Maybe there's some other issue that we don't know about. Okay? But almost perfect results with this Russell algorithm. How much time do I have for applications?
4 minutes.
Okay, really quick through the applications. So I started this lecture by saying, I'm a biologist. What I really care about, and I'm a dry biologist. I use these types of tools, but what I really care about is the applications in biology. So what are the applications in biology? I'm just going to touch on 2 of them that I care about. One is, again, if you haven't seen this before, I will tell you that taking a drug to market is a terribly expensive process. It's really, really, really hard. And so the pharmaceutical companies absorb this huge cost, and then patients who need drugs can't afford them because it's also expensive. It's a terrible thing. So can we learn a little bit? Can we characterize drug mechanisms more deeply, in more detail, and quicker and less money? That's the goal. And the way we're going to do that is by looking at this PGM and adding in the drug nodes. And the way we can do that, remember my multi-well format, we can take some tissue of interest, put it in the wells, and add these different drugs and characterize their signaling network as best we can. And one thing that we are interested in usually is where does a drug hit in the network? Tons of money is spent on asking that question. So we're just going to augment the data that we have. Here's our data matrix. We're just going to add in, this cell was measured under drug 2, and this cell was measured under drug 1. It doesn't have to be binary. If you have multiple doses of the drug, and we sometimes do, you can add in that information. So here's a quick result, drug targets in T cells. This is the canonical, well-known network. Not perfect, but it's what the biologists agree on, roughly speaking. And this is what we get with the Russell algorithm. This is what we get with the standard Bayesian network. I'll just tell you, so you don't have to stare at this, that this is perfect, perfectly represents what we know in the biology. And this one is kind of a mess. It has a bunch of edges that are known, that are thought to be wrong. Furthermore, this is a particular inhibitor, and it hits P38. Now, here's the network. Here's the canonical network. So where should we see it? Well, we really shouldn't see it at P38 again, because we're blocking the activity of P38. So where we should see it is in the targets of P38. And in this canonical network, I didn't show you any targets to P38, so it's not clear where it should be hitting. And where we saw it hitting was at PLC gamma. And so we looked in the literature, and in fact, there is a reported feedback loop from P38 to PLC gamma. Does that prove that our results are correct? No, but it helps us have more confidence in them. Okay? Okay, so the issue most close to my heart is this issue of profiling patients. I'm very interested in cancer. I'm very interested in personalized therapies in cancer. We have a clinical trial ongoing right now, well, that should start sometime soon, where we take children with leukemia. They have failed all rounds of, they have failed multiple rounds of treatment, and these children have a very, very, very small chance of survival, almost vanishingly small. So we take their tumor and we profile it to death. There are other chapters that are going to do other kinds of profiling. We're going to do this kind of mass cytometry profiling. And then what can we offer them? How can we help them? I argue that the best way to help them is to characterize their tumor as well as we can, and then help the doctors to choose which drug that blocks where in the network might help them the most. Okay? So here's the protocol. You take bone marrow or blood from the patient. You put it in this multi-well format. You perturb it however you like to perturb it. If you don't perturb it, you might not see what the network has in store for you. Okay? That's why we perturb them. And then you put it in the test tube with these metal-labeled antibodies. Here's the cytoph again. And you get this, this data matrix. And you might do this for multiple patients. And if you have historical frozen down tumors from years ago, you can also start asking, what does the pattern look like for those who responded to chemo, for those who didn't respond to chemo, for those who survived, for those who didn't survive? So here's a small example using old flow cytometry data. And in it, we were trying to figure out how are the patients different. We took a bunch of patients who had identical diagnosis. And we look at their signaling, and we say, can we find differences even though there's identical diagnosis? And so again, we just add this cell came from this patient, this cell came from that patient. And we get something like this. You can see that they all kind of look the same. But they still will help you see that the correlation here between P38 and ERK has been shifted by the disease state. But we said, OK, this is kind of a messy situation. All the patients look very, very similar to each other. I think the disease state might be obscuring what's happening in the individual patients, because the disease state is causing so much differences that it's overriding the individual patient differences. So we also add in a disease state node. We also had normal, healthy control cells in there. Oh. Sorry, that arrow is not supposed to appear yet. Well, in the interest of time, I'll rush along. But the point was that the Bayesian network here is quite distinct among the patients. And what it allows you to do is to automatically pick out, oh, this joint distribution is different from this joint distribution. And these patients actually look quite distinct with respect to their joint distributions with just these 3 proteins that I'm showing. And that can be very helpful in the context of a clinical situation where the doctor looks at the patients and says, all I know is that they're all kind of the same. And now you're able to tell the doctor, no, actually, this one has a difference in his signaling network over here. And this one has a difference in his signaling network over there. And can you find what's called a targeted inhibitor, which is something that perturbs the signaling network, to treat that patient? Okay? So that's where we want to go. This is using old flow cytometry data. This annoying arrow is telling you that we're now doing it with mass cytometry data where we have many more markers. Again, it's in leukemia, which is a specific one called AML. It's pediatric. We're using 18 patients. And of course, we're hoping to scale up to more. Aaron is the collaborator who made that data set. Also, as a single cell, just one final point. I have a minute. I harped on the benefits of single cell data. Right? So I went on and on about the benefits of single-cell data and how it's important because I can look at multivariate interactions and I have a large data set and it's a big matrix instead of one little vector. But what I didn't talk about is what's important for that for cancer. So in cancer, we think that cancer evolves from a genetic mutation that arises randomly in a single cell. But often cancer is quite genetically unstable. And so often the tumor cell, the initial cell, will evolve and split into multiple subsets. And it's sometimes thought that there's the notion of a cancer stem cell. The different subsets within the tumor have different ability to sort of cause disease. And if you're eliminating only one subset through chemo, or through whatever treatment you're giving the patient, then you're risking you have the risk of the other subsets coming back, what's called relapse, and killing the patient. So we're very interested in characterizing the different subsets of the tumor. You have to eliminate all the different subsets, or at least all the stem-like subsets, in order to save the patient and avoid relapse. How can you do that when you take your tumor and stick it in a blender? So the vast majority of technologies use that. It's not that you absolutely cannot, but it's much, much more difficult. This technology lends itself much better. So here's the tumor here's a cartoon of a tumor. It has different subsets. Obviously, we care a lot about single-cell data for that reason. And so for that reason, I'm just going to touch on this point that if you care about the subsets, now you have to start asking, OK, well, I want to partition. I have this big matrix. I don't know which one came from which subset. I don't even know that there are subsets, and I don't know how many there are. So another thing that I'm not going to talk about, it's not really related to causal learning, but just to tell you guys, because I find it interesting, is this question of clustering or subsetting, separating out our data according to some kind of space. We might use surface marker space, those receptors. We might use signaling space. We might use cell cycle. Different things that might be interesting. And for the AML data, we just started this analysis, so this is very preliminary. But for example, we might find subset-specific signaling that only exists in some part of the tumor and doesn't exist in other parts. And in that particular data set, I can also say, what about the kids who do relapse and the kids who do not relapse? And in a very, very preliminary analysis, we're able to pull out certain links that we find exist in subsets in the relapse versus the not relapse patients. OK, so just a few words on who did this work with me. I'm working with Gary Nolan in the lab. And the main collaborators on the work that I showed were Solomon Itani, a friend of mine from grad school, controls engineer, who also worked with Claire Tomlin, who's currently at Berkeley. And that's it. Thank you, guys. I'm shocked that I got through most of the material.
I think this is a good question. The Wrestle and other kinds of algorithms that you were showing there, the graphs you were putting up looked like they were dynamical systems, but if I understood correctly...
No, they're Bayes nets. Sorry.
The ODE graph you put up, I assume, is a dynamical system.
Of course, yeah.
But the data that those algorithms are getting, that's not dynamical data? Or
OK, so I really rushed through this. The way we were using the dynamical systems to give us synthetic data that mimics our real data. And so what we did is we took, let's say we wanted 1,000 cells. We simulated the ODE model for 15 minutes. And each cell was a different set of initial conditions, protein concentrations. OK, so we randomly sampled a set of initial conditions. And that's cell number 1. And we run it for 15 minutes. That's cell number 1. We randomly sample another set of initial conditions, protein concentrations. And we run it for 15 minutes. And that's cell number 2. And we repeated 1,000 times.
So you do have time series?
No. So this is, we only took one time point. Actually, we had time series, but that was only to compare.
Snapshot after 15 minutes.
Exactly.
Okay.
And that's to mimic the data that we have in reality. We also did a little analysis where we said, how's it going to change if I look at 10 minutes, 15 minutes, whatever, 17 minutes, because we could do that easily. And the answer was it didn't change much. It changed a little bit, but not much.
Question. Why do you need to use networks versus supervised learning to classify patients?
Okay, we also use supervised learning to classify patients. But the reason is that we're interested in characterizing the network as much as we can in order to learn as much as we can about the underlying biology. But we absolutely also use supervised learning. This is a causal meeting, so I didn't talk about supervised learning. I talked about what we can learn about the underlying causality of the system. But yeah, no, that's absolutely another thing you can do. 1 second. The supervised learning will sort of do feature selection for you, depending on what you use. Here, we're interested in understanding the networks because we know we have a lot of hidden variables. We don't always know what they are. And we're very interested in being able to tell the doctor, here's where I think you should perturb, okay, in order to try to save this patient. And since that's our goal, we're more interested in getting as much detail about the underlying biology as we can.
When you fit your graphs, do you regularize in any way, or is there a tuning parameter? And
yeah, so the BDE score, which is the Bayesian network scoring method, it has sort of a built-in regularization penalty.
And you just use that one, or you tune
Sometimes we sort of cheat and add in a bit more regularization because our data sets are quite large, right? And a nasty little secret is that they're not really IID. I mean, you know, these cells are all hanging out together. They affect each other. So because of that, we feel like we have sort of inflated scores. And so sometimes we add another little regularization penalty. But the BDE is not bad. All right.
All right, thank you so much.